phylogenetic tree describes the evolutionary relationship between a set of species. Each species is represented in the tree by a leaf. The inside of the tree represents the evolutionary branching process that produced these species. A node is a speciation event. It represents the most recent common ancestor between the set of its descendants. The length of an edge in the tree records the dissimilarity between the genes of species. The shorter the distance, the more genetically similar those species are, and the longer distance indicates that more mutation has occurred. Say, for example, that we have 5 bacterial species for whom we've sequenced a series of gene sites. If we build a tree describing the similarity between these gene sites, we'll end up with a phylogenetic tree for each gene site and a set of trees overall. These trees are unlikely to have the same shape. In phylogenetics, this is called the tree's topology. From this set of trees with different branch lengths and different topologies, we would like to find a tree that best summarizes the actual evolutionary history between the 5 species. One standard and intuitive way of doing that would be to define something like an average of these gene trees. Looking at these qualitatively very different trees, it's not immediately obvious what the average should look like. In order to find it, it's going to help for us to be able to measure the distance between any 2 trees. In order to do this, we're going to construct a geometric space of all possible trees, called tree space. And this is going to consist of trees that can be built with a particular set of leaves with any shape and any branch length. So let's say we have this set of leaves from which we can build, say, this tree. We can represent tree space around this tree using coordinate axes. Each axis will correspond to the length of one internal edge of the tree. So this axis, for example, corresponds to the distance between this node and that node. If we reduce the edge length between these 2 nodes, this node will essentially slide over to the root node, creating this non-binary tree. If we reduce the edge length between these 2 nodes, this node will slide over to the most recent common ancestor with leaf 3, creating this tree. We can see that this tree is no longer binary, meaning it has some degree 3 vertices inside of the tree. Instead of always having 2 branches coming from a node, we now have 3. Biologically, this is unlikely, but it sometimes represents the information that we have. In tree space, these non-binary trees happen at axis boundaries as edge lengths are reduced to 0. By varying the length of these inner edges in trees with different topologies, we find that on the boundary of these regions, some of the trees match. Where they match, we glue the boundaries together. We can do this with large sets of topologies, and tree space is constructed. And when we do this with all of the different tree topologies, this quickly gets difficult to draw. So here, for this example, we can start folding it so that we can represent this particular tree space more easily. But as we add species, even just one more species, the size of tree space increases exponentially. And this is the best we can do. So now we have our tree space, which means we can measure the distance between any 2 trees. And we do this by finding the geodesic. That's the shortest path between 2 points in tree space. So each of these points represents a tree. There is a unique shortest path between them. Sometimes it'll be a straight line if they have the same shape, and sometimes it really won't be a straight line. And this is because the continuous set of all trees, tree space, is intrinsically and fundamentally not flat. It's actually almost hyperbolically curved. Now, most statistical theory is developed for flat Euclidean space, but here the method we're using by relying on geodesics and geometric objects inside of tree space, allows us to adapt Euclidean methods to explore the whole curved tree space. Incidentally, in statistics, and this is related to the curvature of the space, but because tree space is so large combinatorially, many techniques will use assumptions to reduce its size and make it searchable. The difference here is that we can actually use the full tree space to eventually find the phylogeny that we think best suits our data. Going back to our 5 species of bacteria, we can now use these distances to measure the distances between our gene trees and compute the average or Fréchet mean tree from this set, our species tree. If we were to analyze these trees as vectors, for example, our average might not be a tree, but since we're doing it in tree space, the average is a tree. We now have built a tree that estimates the overall relationship between these different bacterial species. So once we have our tree space, this is one thing we can do. There are many other things. We can, for example, consider the bias inherent in different methods that are used to build trees. Or we could find outlier trees, classification, clustering, other data analysis tools. Having tree space and its geometry defined really allows us to see how abstract phylogenetic trees relate to each other in their natural habitat.